Good afternoon, everyone. Thank you all for connecting during your lunch break to hear my presentation. I will start with playing a short video with a brief overview of NMR facility. It's 3 minutes, and then I will talk. Welcome to the Magnetic Resonance Research Center of the University of Notre Dame. We are a facility serving nearly 200 individual users from 40 laboratories across the campus. We are providing you with access to NMR instruments ranging from 300 to 800 megahertz. You can study your samples either in solution or in a solid state. You may opt for fully automated system with minimum hands-on requirement to fully manual systems where you can do lots of sophisticated experiments. In addition to the nuclear magnetic resonance, electron paramagnetic resonance is available on the X-band EPR spectrometer. When you need to find us, first you type in nmr.nd.edu in your browser. You land on our main page and then navigate to NMR training and resources. NMR training request is what you need to fill out. This is how we start. Your basic NMR training allows you to access automatic 400 in McCourtney Hall, manual 500 in McCourtney Hall, and manual 400 in Stepan chemistry. When you need more resolution and more NMR time for better sensitivity, you get additional training to use Varian NMR 600 MHz instrument. If your compound is at a very low concentration, you really need extraordinary sensitivity. Then you go to a Bruker 800 MHz system with a cryogenic probe. This system is also your choice when you have very many samples to measure and very little time to do it. Here is one example. The top spectrum is the carbon-13 signal recorded at the 800 MHz in 1 hour. The bottom spectrum is the 400 data obtained in the same time on the same sample. To make 400 produce the same sensitivity, we need to run acquisition for 7 hours. Now imagine you have 20 samples to analyze in your project. Where would you go? Our main mission is to support your research, make you successful and productive. We are available to train and advise you from the very beginning of your project through the data acquisition and interpretation. With any of your questions or problems, please email us at nmr@nd.edu. All right. So I need to move the slide. Oops. Here we go. Okay. So in my presentation, first, I will remind you what NMR spectroscopy is and what it is good for. I will highlight capabilities and limitations of NMR instruments and NMR spectroscopy. And we'll give you some examples of successful projects carried out in our facility and finish with costs and policies of the core. NMR spectroscopy is a non-perturbing technique. This means that there is no need to chemically modify your molecules to be observed by NMR. Magnetic resonance relies on the fact that nuclei of some isotopes of common elements are magnetic. All NMR detectable isotopes that we use are stable. Hydrogen 1 and phosphorus 31 are naturally abundant, so they are present at 100% in all samples. Carbon 13 is only at 1% of natural abundance, but still possible to use directly. Other nuclei may be used too, which would be nitrogen 15, deuterium, among others. I didn't mention fluorine as well. And for nitrogen 15 and deuterium, we would need to enrich the samples. What is important that chemical and biochemical properties of your molecule are affected by isotopes to a very small degree. This is why nuclear magnetic resonance is so attractive. We observe molecules as they are, without attachment of special chemical groups to allow for detection. Size of the molecule matters a lot for NMR.
We call compounds with 1 kilodalton molecular weight or less small molecules or small to medium-sized molecules. These are chemical reagents, reaction products, natural products, metabolites, synthetic drug candidates. For these, we typically would observe proton, carbon, and phosphorus natural abundance. You can determine molecular structure, access purity, contaminants, lot variation. You can also directly monitor reactions, chemical or biochemical reactions directly in the NMR tube and observe structure reaction mechanisms and structural changes. We can separately detect components in a mixture based on the different diffusion coefficients. We can also separate spectra by fitting to separate spectra of different compounds. Complex natural products typically come at low concentrations, and this field particularly benefits from use of our 800 megahertz system with a cryogenic probe. High molecular weight molecules also, like peptides, proteins, and nucleic acids, are also doable with the 800 megahertz system. When we use peptides, up to 2-3 kilodaltons, they actually may be done at natural abundance without a need for isotopic labeling. Proteins must be isotopically enriched, and detailed studies are possible up to about 30 kilodalton. With specific method labeling, molecular weight for detection of proteins which may be used is going up to 100 kilodaltons or higher. Every method has its limits, and major advantage of NMR is that it interrogates nuclear spins, not electrons, and therefore it does not perturb bonds. This major advantage, so this is the plus. This major advantage leads directly to a major limitation of NMR spectroscopy. Nuclear spins are very weak magnets, so they give a very weak response. So this is, therefore, NMR needs a lot of spins in the sample, relatively high concentration of your material. So this is a downside. When you're working with smaller molecules, you don't need isotopes. It's a plus. You may work directly with them. Larger molecules require isotopic enrichment, and they typically give lower sensitivity. So it's a minus. But once you have assignments for larger molecules, you can actually, you can directly monitor ligand binding events, assess mutants, assess binding interactions. And this is definitely a plus. This is all without solving structures again, as long as you have the initial structure of the molecule. So first, I will talk about small to medium-sized molecules, and I will be talking about, to give you an idea of sensitivity on our flagship instrument, 800 megahertz with a cryoprobe. One-dimensional proton detection requires smallest amount of materials. For example, 10 microgram of 300 Dalton size molecule or 30 microgram of a kilodalton size molecule is enough, and you will get a good spectrum within 1 hour or much less, much shorter time. The one-dimensional carbon requires more material, 1 to 3 milligrams, and it would need some 4 hours for data collection. This is because magnetically active carbon-13 is only 1% of natural carbon, and its nuclear spin is 4 times weaker than the proton. Two-dimensional experiments relating proton and carbon, the HSQC and HMBC, with 3 milligram of sample will need 8 to 16 hours acquisition time. However, if you can double the quantity, your acquisition time drops by a square of that factor. So if I double the quantity, I reduce acquisition time by a factor of 4. I will start with the simplest application of NMR spectroscopy. NMR signal area is directly proportional to concentration of compounds in a sample. Peaks from different compounds are often well resolved in a spectrum, so we can quantitatively measure purity of particular samples or compositions of the mixture. For example, one of our users scales up a biochemical protocol They worked it out with a high purity reagent from sigma and now want to make more material. Reagent now comes in a larger size from a less expensive supplier. But is the purity still acceptable? I instructed them to make a very concentrated sample, 100 millimolar, to be able to see minor contaminants. Here are the proton spectra of 2 different samples and a reference. So we have sample 1, sample 2, and a reference trace with assignments. Contaminants are shown with the red arrows, and we see a few of them. And very first thing we noticed that sample 2 is by far the cleanest. It's the cleaner than sample 1 by very large degree. We can, looking at sample 1, we can directly determine quantity of each contaminant. First, we will create a scale. We will integrate a known peak, which is this peak. And we know that there are 2 protons in there, and that's the peak of our compound at 100 millimoles. And then when I integrate these 2 contaminants, I see that one of them is at 1%, the other one is at 0.25. And this is a directly quantitative information from the NMR sample without separating it. What were those compounds? So in this particular case, we are identifying peaks of the contaminant in the actual spectrum. Everything else is cut out, so the spectrum seems very clean, but it's just the 2 peaks of the compound. And it was, we proposed that it was an alcohol they used for in one of the steps of purification. So what we can do, we can take a formula, molecular formula of the structural formula of the compound and simulate its spectrum. So we are seeing that these 2 spectra are very similar. They have same splitting and very similar chemical shift. So this contaminant is most likely an isopropanol, and we didn't do any separation. It's just the very same sample which we prepared initially. What if? So you may think about purity test as a routine test for any organic compound you can use. It will be used in the laboratory. You can verify how it is, how clean it is, and how purity changes from lot to lot. It's not unusual then in biochemical protocols when you finish one bottle and start a new one, your results suddenly become slightly different. And this is easy to check with NMR. So what if it is not purity, but identity is in question? Is it actually the compound we think about at all? So very simple approach with NMR is to collect spectra, proton and carbon spectra, and the frequencies of all spins appear at specific positions, and they create some sort of a pattern, which you may think of as a fingerprint. Even without assignments, you could use that pattern to see if it matches the compound. So in here, we have an experiment where we collected the carbon spectrum, and this is the molecule which we thought it is. We predicted the carbon spectrum for this compound, and if you look, apart from some changes in the chemical shift, the pattern is pretty much matching. So this is a very strong evidence that the compound is what we think it is. We always suggest to do mass spectroscopy along with NMR because it gives you another dimension, which helps you be more confident. What if we have a compound we don't know structural? So we need to do a de novo structure determination. In the past, it was a purely NMR expert area. And today, with moderate complexity molecules, basic NMR expertise is enough because with help of a user-friendly software. So there are 2 general steps in structure determination, general phases. One is determination of connectivity of carbons, essentially establishing carbon skeleton. And the other phase is determination of configuration of optical centers and rotamers of rotatable bonds. So we are licensing software from Mystery Lab Research, which is called Domnova, and it has plugins, which is structure elucidation. It helps with carbon skeleton, and we have it, and I will demonstrate how it works. We also have a stereo filter plugin, which helps to determine configurations in rotamers. We're considering buying it at some point in the future. So if I want to do a de novo structure determination of a compound, first I start with acquisition of NMR data. I will collect proton and carbon one-dimensional experiments, process spectra, and identify resonance frequencies. We call it peak picking. Second, I will obtain two-dimensional correlations, essentially correlating protons in a molecule with carbons in a molecule, and every connected pair will give me a cross peak. So this is an example HSQC spectrum, and there are also HMBC and other correlation spectra as well. So once we have peaks in this spectrum as well, we can, once we have the peaks picked, we can submit this data to structure elucidation, a NOVA structure elucidation plugin, and it will try to find all possible solutions to how these carbons may be connected to these protons in a particular structure. So in this case, a molecule was very simple, and I had a lot of information in these NMR spectra. So in 23 milliseconds, I got the structure back, which is menthol, and indeed it was a standard sample of menthol, which I put in an NMR instrument. The very important part is that this is not just one solution it ran into. It creates a whole family of solutions. And if your data is unambiguous, one solution really stands out, like in this case. If your data has some ambiguities, you will have alternative structures to consider. And this is where you will use your chemical and biochemical and NMR spectroscopy expertise to push this forward. This slide shows an example of a structure determination of a natural product, which was done at University of Notre Dame. And here they used cryoprobe on 800 megahertz system. That's the only way forward with natural products, because they're always coming in a very small quantity. Oftentimes they're unstable, so you cannot allow for acquisition being too long. The publication time for this structure was 2012, so they did not have a NOVA structure elucidation, and all the work was done manually by an NMR expert. When molecules are like this, or even more complex, even with a NOVA structure elucidation, the task becomes non-trivial. So if you have a project with significant involvement of natural products and you will want to be confirming structures, it's better if you establish a collaboration with some NMR laboratory who will be helping you to get to speed. We are able to help you acquire all necessary data, train you on processing and using NOVA structure elucidation here at Notre Dame. Okay, what if my sample is a mixture? If it is not just one compound, but several, and it's one solution, I don't want to separate it, I don't want to touch it. It contains all the compounds, and I want to know about them now. So an NMR spectrometer can record spectra of compounds as a function of diffusion coefficient. This is called DOSY, or diffusion ordered spectroscopy. Here on the x-axis, it is just normal chemical shift, and you see a proton spectrum on top with corresponding peaks. The y-axis is a logarithm of diffusion coefficient. Smaller size molecules, so faster diffusion rate, the peaks appear on the lower part of a graph. And everything moving slower appears closer to the top part of the graph. Essentially, this method distributes the peaks you see in one-dimensional spectrum in these two-dimensional plane. For example, this molecule is a benzene. It's our solvent, which we have, and we know it's going to be moving fast. The compound shows these peaks, and we have them all appearing at the same diffusion coefficient. It is indication that it's one molecule, and actually now we have a spectrum of just this compound. There are 4 more peaks in a spectrum which belong to other molecules. These are contaminants 1, 2, and 3, and at this moment we didn't bother to figure out what they are. But we certainly know they're separate. These peaks do not belong to the compound of interest. We can run DOSY representation is very easy to obtain for any sample, and you may record proton spectra, carbon spectra, fluorine, phosphorus, DOSY, So you may think about this as a non-destructive analytical tool for complex mixtures. If we keep going up in molecular weight, so our next stop is peptides. As long as peptides are not too long, less than 30 amino acids, it is still possible to work with them without labeling. So you can use you can look at natural peptides, peptides coming from complicated synthesis where you cannot put in isotopes. We're going to be using proton and carbon at natural abundance. Still, 800 megahertz with a cryoprobe is a must for this type of work, and concentrations should be significant. This is an example of a work done at our facility by a user. They were able to confirm the sequence of the peptide. They determined its helical preferences in solution, and this is a representation they made that it creates helices at certain points in its sequence. However, having said all of that, this type of analysis is already quite complicated, and basically here it requires attention of a full-time NMR spectroscopist to be able to complete this type of project. Proteins. These are much bigger things, and isotopic enrichment is a must, and also highest field available with a cryogenic probe is also a must. So our facility uses 800 megahertz system with a cryoprobe for all protein work. And for proteins, isotopic enrichment allows to put in an isotope label in pretty much any part of a molecule. The simplest and the cheapest labeling pattern would be with nitrogen 15. And this labels all amide groups. Protons are naturally magnetic, and nitrogen 15s are going to be paired with these protons. So this would be a depiction of 22 kilodalton molecule with all NH groups shown as spheres, which are showing up in the NMR spectrum. Essentially, entire structure is being uniformly covered by this type of labeling. So after data acquisition is complete, we have these correlation spectra, and this now is an expert area. This is where you need to have a dedicated researcher in the laboratory dealing with this type of work. We can train, but it's a lot of work for someone dedicated to the job. So what you can do with this is that, of course, you can attempt structure determination. It is not an easy task, and in most recent times, it is easier to obtain structure by X-ray and then utilize it for interpretation of NMR spectra and in NMR data, because structures differ very little between crystal and a solution in most cases. When you have a structure, you know what signals come, where each of these peaks coming from in a molecule. So if you can do detection of binding interactions or conformational changes, you can determine binding mechanisms. So I will talk about binding interactions then. That's my particular area of specialty. I spent significant time working with this type of method. And basically, as I said, you have a protein labeled with, in this case, it's NH. These are NH groups, nitrogen 15. This is the cheapest labeling of any recombinant proteins you can have. And when we have these signals detected, it gives us this two-dimensional correlation map or spectrum. Now, if I'm adding a ligand, it attaches to a particular part in the molecule. Here it is in this binding cleft. And specific sites surrounding this molecule respond. They get affected. The chemical shifts change, and this is what we see immediately. So even without assignment, you can determine that that molecule binds to a protein. If you have an assignment, you know which residues respond. If you have a structure, you know how they are arranged in space, and you may begin thinking about a mechanism. So NMR data are not just these spots with frequencies attached corresponding to them. These peaks have a shape, which we call traditionally a line shape, though it is a peak shape. And this is the depiction of how the peak shape changes in a titration experiment where I'm adding a ligand and peaks shift. So this would be a two-dimensional representation where we have a peak at an initial position, and then when his ligand is added with half equivalent, 1.5, 3, the peak shifts to a new place. This is how we saturate the binding site. And if we look from the side, the peaks actually change shape pretty dramatically. And this is what may be used to extract information about the mechanism. If it is just a simple one-to-one binding mechanism, like lock and key mechanism, nobody gets surprised. You can expect that. But if the mechanism involves some sort of conformational changes or like induced fit or some other interactions, this becomes a complex mechanism, and it is possible to get an idea that it is happening in this particular molecule. My last example of what you can be doing with NMR spectroscopy is analysis of reaction kinetics. It may be a chemical or biochemical reaction, it may be enzyme catalyzed reaction, it may be just a direct reaction between components. For NMR, it doesn't matter. As long as you can record the one-dimensional spectrum, and as long as a reaction is slow enough that you can follow it with NMR, you are in business. Slow enough. It's a relative term on a regular NMR spectrometer where you don't have any type of fast mixing apparatus, as we don't have. It is the time scale of minutes, hours. So you need to have a reaction in that times occurring in that time scale. If somebody uses a fast mixing apparatus, the reaction time scale shifts closer to seconds. So here, in this particular case, it was hours, and you can always change temperature or dilute components, so the reaction rate will become slow. It's not that the reaction act itself needs to be slow, it's the conversion rate. So in this case, we are collecting a one-dimensional spectrum which gives us all peaks of initial substrate, initial reagents, and then they convert into products. For example, here there are no products yet, and these are the products. We collect this bunch of spectra, and we need to now analyze them to extract what happens in this reaction mechanism. For analysis, it is important to have a model. So you need to have an idea or hypothesis of what kind of process occurs in your reaction, and you may have multiple candidates. This is all possible to test. We are able to determine whether we can fit kinetic rate data with your hypothesis, whether it is supported by the data, and which hypothesis is better support, and what meaning of these particular rate constants for reaction dynamics. So this is one example where our collaborator went overboard with very complicated mechanism, which is basically where we have two reactants and multiple molecules of them making a conglomerate intermediate, which then splits into a final product and one of original reagents which feed back into the reaction, beginning of a reaction. And this would be represented by a pretty complicated set of differential equations, but it's all taken care of. As long as we can derive the mathematical form of the equations, we can plug it into software. So in order to get all of this happening, I wrote a software program which supports this data fitting, and it's available free to anyone in academia with academic license, and it works in MATLAB. So if you are interested, we can talk about it and we can collaborate. So this is just a general workflow for that kind of analysis, where initially you start with acquisition of NMR spectra, integrating peaks, and extracting them as these traces. So now these traces kind of don't have relationship to NMR. They might have come from somewhere else, but very important that in NMR spectrum, we have very many peaks separated well enough such that you can monitor essentially all of your components of a mixture, all of your intermediates separately. It's as if you were separating reaction mixture at every time point, which we don't need to do in NMR. So we have these reaction progress traces. We put in the model. We try to fit. We see if it fits at all or not, and what fits and what kind of models fit similar well enough, we can do statistical analysis to determine which one of them actually works well. And then once you have the rate constants, that's not the end of the story. We can now model the reaction dynamics at different concentrations, because think about this. You are studying a process in a test tube to be able to observe it, but in reality, it probably occurs in biological systems at very different settings, in very different concentration ranges. So you will want to simulate the reaction progress which corresponds to conditions of actual natural process. All right, so I'm done with my science stuff, and I'm going to just give you a brief overview of how we operate. NMR Center at Notre Dame is subsidized by Department of Chemistry and Biochemistry to a very large degree. 2/3 of entire budget is paid by department. So our users enjoy relatively low NMR fees. So that's an example of a fee, like $10.40 per hour for 800 time, and for 100 time would cost approximately twice as little. We do not charge for training or basic consultation of instruments and software. So we are interested. So basically, our goal is to make you a confident user. And in some ways, it's self-interest because we're very short-staffed. We have only 2 people in the facility. So we're doing best if we're able to train you and you are doing your project with our consultations and help, but it's all in your hands. In order to help you do it, all in your hands, we created a remote access capability to our spectrometers. We have sample changers where we can line up samples and you can access the spectrometer from your office or even from off campus. We have TopSpin available for everyone on campus. It's a software which comes with Bruker spectrometers. We are licensing MNOVA NMR, which is available to all users of Notre Dame campus. And if you are not from Notre Dame campus, you are welcome to use our workstations on campus, which have these licenses on them. All right, and very last thing, which is probably not the least, once you're done with your work, we are offering free service of editing your results and methods sections regarding NMR to make sure that papers include best presentation of NMR data, most professional presentation. I think I am completely done, and please go ahead with your questions. I think I see something in the chat. And okay, so go ahead, type in your questions if you are interested to hear about anything else, or basically, and I'm typing in nmr.nd.edu is the website of our facility, which I mentioned in a movie, and this is where you can go and get in touch with us.
Great, thank you very much. While we're waiting for a question, I have just one. Just thinking about if someone from Purdue or Bloomington or something wanted to use your instrument, would they basically bring their samples up and use it, or could they ship it to you and you would run it, or how would that work the best?
So they can do either way. The preferable option for people who are really off campus, you need to come at least once or twice to get trained, because what we will want to do, we will want to be make you able to operate the spectrometer remotely. We will be shipping your later samples to us. We will be putting them in a sample changer. Sample changers have cameras, so you can actually see what is in a sample changer, and you operate spectrometer directly from your desktop computer. But you will need some expertise in order to operate it. So you will need to be trained first. So this is the way of operation for remote users. If somebody has a really burning project, which is really like grant proposal due 2 weeks from now, there is no time to train, we can do some limited service. So if you send us a sample, we can I can actually just set up experiments, run them for you, process them, ship the data back to you. This will cost some $60 an hour. So it's not very difficult to do. It's just my time is short. So we do it in kind of extreme cases when people really ask, we really need this done now. But otherwise, there is a remote connection. There are sample changers on every instrument, and I can be putting samples in for you, and you will be able to run them. And basically, at some point, I guess I will be shipping samples back to you when everything is done.
And then is there like a sign-up sheet, or like what would be the wait time if someone had a project they were ready to do?
Okay. For anyone from outside campus, I would suggest you talk to me first. Well, you will always talk to me first, because Notre Dame will want to establish you as external user, and it will be a little bit of an ordeal. There will be signing of a paper involved. But you talk to me, and we can book the time appropriately. I can tell you at this time, we have enough open time slots on instruments pretty much for immediate use. It may be difficult to find time if you tell me that you need time today, but still often possible. And since instruments are maybe operated remotely, they may be operated at night, they may be operated on the weekends, and you, generally speaking, don't need to show up for that. Unless something gets stuck, then you contact me, I will show up, look at the instrument, and it will get fixed. I want to mention one more thing. We can train remotely as well. Our software allows multiple connections to this to the desktop of the instrument. So you see a desktop of the instrument and you can operate it, and I see that too. So it is pretty much as good as sitting next to each other, but it's better because each of us has our own keyboard, and we don't really need to crowd around the screen. So there are multiple different ways, but basically there will be some amount of effort, more effort than if you are on campus. Of course, it's better if you are on campus. If not, still possible.
There's a question in the chat. Do you provide training or tutorials to run TopSpin?
Yes, we provide training and tutorials, and we are continuously developing them. I'm typing, so if you go to NMR.oh, actually, I can go on my screen. I should be able to just say, so this is nmr.nd.edu. This is our website. So if you go to training and resources, there is this instructions and tutorials link, which takes you at this time to external website, which is still our website. And here you find first, this is our general training course. You can take it without signing up because these are just videos. And it tells you about all the policies, all the ways the facility operates, safety and ways of booking time, ways of sample handling, and a required protocol for safety we need to follow. So we have data processing in TopSpin is covered to some degree. We are covering new topics as we get requests. So some topics are covered comprehensively, some less comprehensively. Our main, when you get basic NMR training with us, you also get a big video of recording of how you operate Bruker spectrometer. Basically, just step by step, you just watch through it and you can do it yourself too. So this is kind of a training part when you can just click on it and find instructions, for example, how to run COSY experiments. This is COSY, and any two dimensionals in our facility start with training on COSY. And generally speaking, it is designed such a way that people watch it, and then they are able to go to spectrometer and do it. So I'm not involved at this point unless they run into trouble. But since you can watch it on your mobile device, you can be you can open it on a phone next to a spectrometer and just follow the steps. So, and then once you're done with COSY, we would go to HSQC, NOISY, whatever else two-dimensional, because much of it is covered in COSY. We are going to be just talking about differences. So this is training for special experiments. And when you are at any particular spectrometer, basically just scroll down. Every particular spectrometer has a page where it now lists instrument-specific things, because general stuff applies to all of them. Instrument-specific stuff is covered here. Basically, I'm saying first things to do, what you have to do on this machine. This machine is equipped with sample case. So you need to go through sample case training, and it's again also video-based, basically, and you have instructions as well. You don't need to watch video again if you need to just review it. So every time I have actual instructions. And if there are problems which occur in spectrometers, first time people connect to us. Basically, they email us. We watch our emails pretty much 24/7 as much as we can. And if emergency situation happens, oftentimes you're able to do it yourself with our guidance. So this would be a guidance for rebooting the 400 megahertz system. Basically, telling you what to push, what to open. Here is the button to push. Here is the window to look at. So so far, 100% of our users who were getting stuck in the late night at the instrument were able to reboot it with just me sending them that link. And people who did it already once are able to do it again without talking to me, just telling me later, spectrometer got stuck, I had to reboot it. So we are trying to be able to respond as quickly as we can. The system on every spectrometer is such that when you finish your work, there is a questionnaire which you have to fill. You say what you used, what sample was there, and if there were any problems. So our advice to everyone who gets issues, even if it is not a spectrometer issue, but you just forgot something, you log out of the instrument, and in that form, you put in your question. The instrument automatically emails it to me and our engineer, Justin Pontis. One of us sees it, and we can connect through Zoom pretty much immediately. So oftentimes we're able to rescue the situation straight while you're at the spectrometer. Otherwise, if not, we are booking time later and meeting with you. Most of the time, again, we are meeting on Zoom because we have instruments in 3 places, and running between buildings is not very effective. So it's actually faster for me to book the time for training, and then you show up at the spectrometer, and we connect through Zoom, and I connect to your workstation, and you are able to do it. If you are off campus, you don't need to show up at the spectrometer. We book the time, you connect to the workstation, I connect to the workstation. As long as your sample is in a changer, we can put it in. So we're trying to make it as contemporary as possible in terms of communications, and so far it works. Any other questions?
Yeah, that's a fantastic website that you have there, with very comprehensive. I think that should be very helpful for people. Thank you for showing it. Does anyone in the audience have a question? You're welcome to unmute yourself and ask. Well, it looks like there's no questions. I think you're very thorough. So I want to thank you again for presenting and everyone for attending.
Thank you for listening.